Carry on.
Okay, okay, folks. Today let's get started. Hope you are all doing awesome and in a perfect vigor as well. This is Raja Ahmed, a junior year lad pursuing computer engineering from UET Peshawar, Pakistan. Communities are home to cutting edge advanced technologies. We believe in peer to peer learning and always raise the flag of community culture. I being the lead at prestigious communities of UET Peshawar, Google Developer Student Clubs, and Microsoft Student Ambassadors Club UET Peshawar. Today, we have a lot of amazing folks from all around the globe in collaboration with us. We have Mr. Asharib Ahmed, the DSC lead and the Pi and AI ambassador from SMIU Karachi, Mr. Ahmed Raza, the Pi and AI ambassador from SMIU Karachi, NG Alieva, the DSC lead from Oil and State University, Azerbaijan. And then moving forward, we have Mr. Shehzad Umar Beg, the Microsoft Student Ambassador from SMIU Karachi. And last but not the least, we have Mariamavit Amnatu, the DSC lead of the Debre Behran University. So it's an amazing sort of collaboration from an awesome pool of talented folks from all around the globe. So today we have our very hot and trendy topic, that is artificial intelligence in healthcare. And today we have our very renowned personality with us, our guest speaker. We all welcome you virtually, Mr. Joshua Starmer, the founder and CEO of StatQuest and board of director at Society of Scientific Advancement. We wholeheartedly welcome you, Joshua.
Thank you very much. Let's get started with a silly song.
So let's get started. And in order to have an introduction of our leads going forward, I would like to invite Mr. Shah. Please introduce himself and his chapter as well.
Okay. Thank you, Rajama, for the amazing welcome. First of all, I want to thank you, George Thomas, who came today for this event, and all of the organizers who put their efforts together for bring this amazing event for you guys. And my name is Shari Ahmad. I am Developer Student Club leader of St. Madrasa Islam University, and I think that it is enough now, and we will go forward.
Moving forwards, it's over to Shehzad. You can introduce yourself and your chapter as well.
Uh, Hello, I'm Shazad. I'm just the similar classmate of like Asharib, and I'm not kind of a bigger AI passionate person, but that's it.
We have Inzi Aliyeva, the Google Developer Student Club lead from Azerbaijan University. Over to you, Inzi.
Hi everyone, thank you. My name is Inci. I'm studying computer science at Azerbaijan State Oil and Industry University. As you know, I am also a team lead of Developer Student Club at the same university. Thanks.
Going forward, we have Ahmed Raza, the Pi and AI ambassador from SMIU Karachi. Over to you, Ahmed.
I'm Ahmed, Pi and AI and Deep Learning.ai ambassador from Karachi and AI lead in Developer Student Club SMIU and also pursuing CS in SMIU. Thank you.
Well, folks, artificial intelligence and healthcare is a perfect combo. And without wasting any time, so let's get started. Over to our esteemed personality, Mr. Joshua Starmer. Folks, we are determined to present and have you a very wonderful sort of experience. Over to you, Joshua.
Okay. Machine learning and genomics. We're gonna put them together. Hip, hip, hooray, StatQuest. So let me share my screen so you guys can see what I'm seeing. Let's look at this. Hooray. So today we're going to talk about machine learning as the microscope for something called genomics. And genomics is sort of a style of biology where you study the genome or all the genes and the genetic material that kind of create who we are the way we are. So without further ado, I want to say that this quest consists of 3 parts. The first part I will introduce a very simple machine learning method called naive Bayes. And then in the second part, I'll provide a brief overview of a genomics technique called ChIP-seq. And then I'll show you how to combine machine learning with genomics data to generate new insights. And note, machine learning refers to the techniques used for self-driving cars, identifying your friends in the pictures you take, and recommending TV shows. These fancy sounding methods also work in a lot of other settings, including, as we will soon see, biology and genetics. So first, let's learn about a very simple machine learning method called naive Bayes. So imagine we received normal messages from friends and family. And we also receive spam, unwanted messages that are usually scams or unsolicited advertisements. And we wanted to filter out the spam messages. So the first thing we do is make a histogram of all the words that occur in the normal messages from friends and family. We can use the histogram to calculate the probabilities of seeing each word given that it was in a normal message. Excuse me. For example, the probability we see the word dear, given that we saw it in a normal message, is 8. The total number of times dear occurred in normal messages divided by 17, the total number of words in all of the normal messages. And that gives us 0.47. So let's put that over the word dear so we don't forget it. Likewise, the probability that we see the word friend, given that we saw it in a normal message, is 5. The total number of times friend occurred in normal messages divided by 17, the total number of words in all of the normal messages. And that gives us 0.29. So let's put that over the word friend so we don't forget it. Likewise, the probability that we see the word lunch, given that it is in a normal message, is 0.18. And the probability we see the word money, given that it is in a normal message, is 0.06. Now we make a histogram of all the words that occur in the spam. And we calculate the probability of seeing the word dear, given that we saw it in the spam. And that is 2, the number of times we saw dear in the spam, divided by 7, the total number of words in the spam. And that gives us 0.29. Likewise, we calculate the probability of seeing the remaining words given that they were in the spam. Bam! Now, because these histograms are taking up a lot of space, let's get rid of them, but keep the probabilities. Oh no, it's a terminology alert. Because we have calculated the probabilities of discrete individual words and not the probability of something continuous like weight or height, these probabilities are also called likelihoods. I mention this because some tutorials on naive Bayes say these are probabilities and others say they are likelihoods. In this case, the terms are interchangeable, so don't sweat it. In other words, we'll get the same numbers or the same output either way. Now, imagine we got a new message that said, "Dear friend," and we want to decide if it is a normal message or spam. We start with an initial guess about the probability that any message, regardless of what it says, is a normal message. The guess can be any probability we want, but a common guess is estimated from the training data. For example, since 8 of the 12 messages are normal message, since 8 of the 12 messages are normal messages, our initial guess will be 0.67. So let's put that under the normal messages so we don't forget it. Oh no, it's another terminology alert. The initial guess that we observe a normal message is called a prior probability. Now we multiply that initial guess by the probability that the word dear occurs in a normal message, and the probability that the word friend occurs in a normal message. Now we just plug in the values we worked out earlier and do the math. Boop, boop, boop. And we get 0.09. We can think of 0.09 as the score that dear friend gets if it is a normal message. However, technically, it is proportional to the probability that the message is normal, given that it says dear friend. Let's put that on top of the normal messages so we don't forget. Now, just like we did before, let's start with an initial guess about the probability that any message, regardless of what it says, is spam. And just like before, the guess can be any probability we want, but a common guess is estimated from the training data. And since 4 of the 12 messages are spam, our initial guess will be 0.33. So let's put that under the spam so we don't forget it. Now we multiply that initial guess by the probability that the word dear occurs in spam, and the probability that the word friend occurs in spam. Now we just plug in the values that we worked out earlier and do the math. Boop, boop. And we get 0.01. Like before, we can think of 0.01 as the score that dear friend gets if it is spam. However, technically, it is proportional to the probability that the message is spam, given that it says dear friend. And because the score we got for normal message, 0.09, is greater than the score we got for spam, 0.01, we will decide that dear friend is a normal message. Double bam! Now, before we move on to a slightly more complex situation, let's review what we've done so far. We started with histograms of all the words in the normal messages and all the words in the spam. Then we calculated the probabilities of seeing each word, given that we saw the word in either a normal message or spam. Then we made an initial guess about the probability of seeing a normal message. And this guess can be anything between 0 and 1, but we based ours on the classifications in the training data set. Then we made the same sort of guess about the probability of seeing spam. Then we multiplied our initial guess that the message was normal by the probabilities of seeing the words dear and friend, given that the message was normal. Then we multiplied our initial guess that the message was spam by the probabilities of seeing the words dear and friend, given that the message was spam. Then we did the math and decided that dear friend was a normal message because 0.09 is greater than 0.01. Now that we understand the basics of how naive Bayes classification works, let's look at a slightly more complicated example. This time, let's try to classify this message. Note, this message contains the word money 4 times. And since the probability of seeing the word money is much higher in spam, 0.56, than in normal messages, 0.67, then it seems reasonable to predict that this message will end up being spam. So let's do the math. Calculating the score for a normal message works just like before. We start with the guess, then we multiply the probability we see lunch, given that it is in a normal message, and the probability we see money 4 times, given that it is in a normal message. And when we do the math, we get this tiny number. However, when we do the same calculation for spam, we get 0. This is because the probability we see lunch in spam is 0, since it was not in the training data. And when we plug 0 in for the probability we see the word lunch, given that the message was spam, then it doesn't matter what value we picked for the initial guess that the message was spam. And it doesn't matter what the probability is that we see money given that the message was spam. Because anything times 0 is 0. In other words, if a message contains the word lunch, it will not be classified as spam. And that means we will always classify messages with lunch in them as normal, no matter how many times we see the word money. And that's a problem. To work around this problem, people usually add one count, represented by a black box, to each word in the histograms. Note, the number of counts we add to each word is typically referred to with the Greek letter alpha. In this case, alpha equals 1, but we could have set it to anything. Anyway, now when we calculate the probabilities of observing each word, we never get 0. For example, the probability of seeing lunch, given that it is in spam, is 1 divided by 7, the total number of words in the spam, plus 4, the extra counts that we added. And that gives us 0.9. Note, adding counts, the black boxes, to each word does not change our initial guess that a message is normal, or the initial guess that a message is spam. Because adding a count to each word did not change the number of messages in the training data set that are normal, or the number of messages that are spam. Now, when we calculate the scores for this message, we still get a small number for the normal message. But now, when we calculate the value for spam, we get a value greater than 0. And since the value for spam is greater than the one for a normal message, we classify the message as spam. Spam. Now that we know how naive Bayes works, let's talk about ChIP-seq. But before we do that, what I'd like to do is see if anybody has any questions about naive Bayes. Anybody have any questions about naive Bayes? Otherwise, we'll just move on to ChIP-seq. All right, we'll move on to ChIP-seq. Let's talk about ChIP-seq.
Folks, you can also enlist your questions in the chat as well.
Yeah. So imagine we have a bunch of cells. Oh, this is a biology example. And inside each cell, there's a nucleus. And inside each nucleus are chromosomes. Let's focus on the chromosomes. Specifically, let's focus on these chromosomes. Chromosomes are made out of chromatin. Chromatin is made out of DNA plus histones, which are a type of protein, plus other proteins that we'll talk about some other day. DNA wraps around the histones to package DNA, and the packaging can regulate gene transcription. And what I mean when I say regulate gene transcription, I mean gene activity. So if you can imagine in our body is made out of cells. So, in my heart is just made up of a bunch of cells, and they're called heart cells. My lungs are also created by a bunch of cells, and they're lung cells. Heart and lung cells do different things. And part of how they do different things is regulated by their genes. So there are certain genes that create heart specific cells that do heart specific things like pumping blood throughout our body. And then there are lung specific genes in the cells that are active only in lungs and are not active in hearts. So actually, let me back up a little bit, just so you know, every single cell in your body, regardless of whether it's lung or heart or eyeball or tongue or any cell in your body, they all have the same set of genes. They all have the exact same genome. And the big difference between the different parts of our body are which genes in that genome are active and which genes are inactive. And so when I say gene transcription or regulate gene transcription, what I'm talking about is what genes are active in that specific cell doing that specific cell type activity. So if you have a heart cell, it's doing heart specific something. And if you have a lung cell, it's got different genes that are active that help it be a lung cell, which is all about getting oxygen from the air and getting that into the blood. Okay, so depending on how they are modified, and we're talking about the histones, these are the proteins that the DNA and the genes are wrapped around. The histones can activate or repress genes. And what that means is it can turn on genes for hearts. And in that same cell, in that same heart cell, the histone modifications can then shut down or prevent all of the genes that are used anywhere else in the body, like the lung specific genes or the eyeball specific genes. The histones can shut down those genes and make sure that within a heart cell, we don't get like the lung specific genes to be active. And we don't want that to happen. We want just the heart specific genes. Okay, so lots more can be said about chromatin and how it's packaged, but that's for another day. Today, we just need to know that chromatin is essentially DNA wrapped around histones. To simplify things, let's just use this big blue line to represent chromatin from here on out, but remembering that it's made of DNA and histone proteins. And let these big brown arrows represent genes. And let this green circle represent a histone that has been modified to promote transcription. So this histone has been modified. So if this was our heart cell, this histone would be modified to cause this heart-specific gene to be active. So the arrow on the left side would be active. And let this red stop sign represent a histone that has been modified to repress transcription. So this histone modification is supposed to stop a gene from being active. So imagine the gene on the right side is maybe lung specific or eyeball specific, and we don't need that gene to be active in a heart cell. So in a cell, all kinds of proteins can bind to DNA. This mustard colored thing might promote transcription of this gene. And this pinkish colored thing might repress this gene. And who knows what this green thing is doing? So we can use a method called ChIP-seq to find out what these proteins are doing. ChIP-seq stands for chromatin immunoprecipitation combined with high throughput sequencing, which is lots of big words. And we're just going to go through it one piece at a time. The specific words are not super important. What's super important are the main ideas. And what ChIP-seq does is it identifies the locations in the genome that are bound by proteins. And that's the most important thing about ChIP-seq. So I'm going to say it again, and I'm going to say it in bold. ChIP-seq is a laboratory technique that identifies the locations in the genome bound by proteins. For example, say like we wanted to identify all the regions in the genome bound by the green thing. The first thing we do is use formaldehyde or something like it to glue all of the proteins bound to the DNA together with the DNA. So I don't know if you guys have something called super glue, but when I was a kid, I got some super glue on my finger and it stuck to everything. That's kind of what formaldehyde is for proteins and DNA. It causes them to stick together like glue. And by the way, the reason why we're interested in gluing everything to the DNA is typically when when a protein binds to DNA, it does so transiently. It'll bind for, a very short period of time, do its thing, and then fall off the DNA. And if we want to identify all the locations in the genome where that protein is bound, and if what we want to do is we want to glue it, we want to glue that protein to the DNA. So then we can investigate sort of where it is. Otherwise, the interaction is too quick and we won't catch it. So we're just sort of sticking everything on. So that'll give us time to kind of investigate the genome. Anyways, when we do that, when we glue the proteins to the DNA, that means all of the DNA bound proteins, not just the ones we're interested, end up being glued to the DNA. The next thing we do is cut the DNA into small, approximately 300 base pair fragments. So these are short parts of the genome. So a base pair in the genome is, one of those genetic letters, G, A, T, or C. And so we take a stretch of 300 of those letters and we chop, we cut the DNA at that spot. Then we isolate the protein we're interested in. In this case, we're interested in the green thing using something called an antibody. The black star represents an antibody that is attached to a bead. And this bead, in this example, has a little bit of iron in it, and that makes it magnetic. And that allows us to isolate the proteins bound by the antibody and allows us to wash everything else away. Then we reverse the formaldehyde glue by warming up everything. Then we isolate the DNA by washing away the proteins. And now that we see how to isolate DNA that is bound by a particular protein, let's take a step back. So far, the example has focused on just these chromosomes. But the process glue proteins to DNA, cut up DNA, bind proteins of interest with antibodies, isolate antibodies, unglue and wash away proteins, it applies to all of the chromosomes in the cell. And usually it's applied to a pool of about 6 million cells because cells are tiny. 6 million cells could fit on the, tip of a ballpoint pen. So we end up with a lot of DNA fragments from a lot of cells. Then we prepare a sequencing library by adding sequencing adapters to both ends of the DNA fragments. And if you're not familiar with what sequencing is, don't worry about that. Just know that this is part of the experimental procedure. They do something called PCR and they amplify the library. Then they check the library concentration and then they sequence. And all sequencing is this is the important part, is converts sort of that DNA molecule into the letters of DNA that we know and love. T's, G's, A's, T's, G's, A's, and C's. And then what we do is we align the high quality reads to a genome. So if this is the genome, so imagine we had a 3 chromosome genome, and typically in a cell, chromosomes are in pairs. So we've got 1 pair for chromosome 1. That means we've got 2 copies of chromosome 1. And this is, this is the way humans are. We have 1 copy of chromosome 1 from our mother and 1 copy of chromosome 1 from our father. So we have 2 copies of every chromosome and just 1 comes from each parent. So anyways, if this is the genome, this is a simplified genome, then the first read might come from here. And what I mean by that is that sequence that we've got a sequence that says TT, AGGA. Well, that exact sequence may occur on chromosome 2 at position 3,200,000, something like some big number, right? It's just some position. It doesn't matter exactly what the number is. And the second read, that sequence, that ATACC, blah, 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 that exact sequence may occur on chromosome 1 at another position. So this is what we mean by aligning the reads or the sequences on the right hand of the screen. We're finding out where in the genome those sequences occur. Anyways, so we just keep doing that. And ultimately, we get a long list of genomic coordinates for all of the reads. And that means we usually have between 50 and 100 million reads. And although this list is way too long to go through by hand, we can visualize it with something called a genome browser track. This specific genome browser contains genes and chromosome positions in the mouse genome. I used to work in a mouse laboratory, by the way, so that's my background. That's why I've got mouse on my mind. Anyways, at the top, we have genomic coordinates, which are chromosome names and positions. Then we have schematics for the genes in this genomic region. Below, we have a track that we created from the 50 to 100 million ChIP-seq reads. And this track is basically a histogram of where those reads aligned. And we can see that the histogram or the track is relatively large in one area on the sort of the left-hand side of the screen. But the reads did not really align to many places. There's not a big pile up like a histogram anywhere else on the right side. So we see a lot of reads mapped in this region and relatively few reads mapped in these other regions. This track was made from a control experiment. The control track was made by taking some of the input chromatin from the original ChIP-seq experiment and, without using an antibody to enrich for any particular protein, ungluing all of the proteins and washing them off. in sequencing, aligning, and converting into a track. In summary, the control track uses some of the same input chromatin for the ChIP-seq experiment, but doesn't try to enrich for any particular protein binding. We use the control track to verify that a high concentration of reads in the ChIP-seq track is due to an actual protein binding there, and not because a lot of reads map to a repetitive region or some other weird anomaly. It's just a way of verifying that what we see is what we think it is. Statistically significant peaks are usually represented on genome tracks by rectangular bars. And we can then compare peaks for the same protein in different cell types, like lung versus stomach. We could say, are there peaks? Is there a peak or a lot of ChIP-seq reads mapping to the same regions in lung versus stomach? If they have the same peaks in both cell types, then we could say, well, that protein is used for things in both lung and stomach. If these peaks only occur in the lung, then we can think, well, maybe that this protein has some lung specific function. Or if we didn't know the specific DNA sequence that this green thing is bound to, then we could guess that it was a motif found in all of the peaks. And this is a genomic motif. The larger the letter is, the more commonly it occurs in the locations where that green thing binds. And so the most important letters in this motif are G, followed by something, maybe C, maybe not, because C is small. That means it could be C or it could be something else, followed by two G's and a T. And that's the most important base pairs. So when this protein is floating around and it sees a G followed by something followed by GGT, it goes, hey, that's where I'm supposed to go, and it tries to bind to that piece of DNA. We can also try to determine the functional role of the gene of the green thing by looking at where it binds relative to the genes. Here we see that the green thing binds near the start of a gene, so it might regulate that gene in some way. So these are just some of the things you can do with ChIP-seq. In summary, ChIP-seq identifies the locations in the genome bound by proteins. Double bam. Now that we understand the main ideas behind naive Bayes and ChIP-seq, let's talk about how we can combine them to gain new insights about genes. Now, going back to this big blue line that represents chromatin, remember, these big brown arrows represented genes. And this green circle represented a histone that was modified to promote transcription. And this red stop sign represented a histone that was modified to repress transcription. Now, in this example, it's pretty obvious that this gene could be transcribed because it has a modified histone that promotes transcription. And it's pretty obvious that this gene may not be transcribed or active because it has a histone modification that inhibits transcription. Unfortunately, in real life, the situation is a little more complex. Instead of just having two easy to interpret histone modifications at different genes, we frequently see them mixed up. And it turns out there are a lot of different modifications. And it's not always clear if they promote or inhibit transcription. That said, if we had a few genes that we knew were active and a few genes that we knew were inactive, then we could treat histone modifications like words and create a histogram of how frequently they occur. So before, when we did naive Bayes, we were looking at spam or normal messages versus spam. Now we were looking at words, and we made histograms from words. Now what we're doing is we've got active genes versus inactive genes, and we're counting the number of histone modifications that are near the start points of those genes. And now when we get histone data for a new gene and we don't know if it is active or not, we can use naive Bayes to find out. So now that we know the theory of how this might be possible, let's look at the steps required to make it happen. First, we need some data. When you don't have your own data, one great resource is called OnCode, which has a lot of RNA-seq and ChIP-seq data that you can download for free. So by the way, RNA-seq is just a way of measuring gene activity directly. ChIP-seq allows us to look at proteins bound to DNA, and RNA-seq lets us to see how active the genes are. So in this example, all of my data came from OnCode. For this demonstration, what we will do is we'll classify genes as active or inactive using RNA-seq data from OnCode, which is like ChIP-seq for active genes. Then we will merge the data with ChIP-seq data for histone modifications. And we'll take some of the active genes and some of the inactive genes and put them to the side to form a test data set. Ultimately, we'll use the test data to evaluate how well the machine learning method performed. Now, with the remaining active and inactive genes, we create a naive Bayes classifier, and then we run the test data through the naive Bayes classifier to evaluate how well the classifier performs. Bam. Now, while this example is not exactly groundbreaking, the point is to demonstrate an approach that can be easily be applied to all kinds of other situations in genomics and genetics and biology. So that's it. That's the end, and I can stop sharing. And we can come back. And you can ask me questions, or we can do whatever we want. We can do more silly songs. It's up to you guys.
Folks, the floor is open for questions. You can enlist your queries down in the chat, or you can unmute and speak it up.
No, we can't unmute, so they can raise their hands, so I can unmute.
Surely in that way.
Yeah, there is also an announcement that I have shared attendance in chat, so you can mark your attendance, and after that you will get your certificates.
Someone asked, is it better? I see can I answer a question? Somebody asked if it's better to use deep learning for this kind of problem. And the answer to that is maybe. You don't really know in advance. Deep learning is fantastic, and it's great. It's not always the best solution. One thing that's nice about naive Bayes is it's relatively easy to see how it's making its decision and sort of see what the major players are, which is something that's sometimes very difficult to do with deep learning or neural networks, right? Neural networks are more of, I mean, a very simple neural network is not a black box and you can see exactly what it's doing. But if you have a bunch of inputs, enough inputs and enough outputs, it becomes something where you can no longer see what it's doing. And so it can make, the neural network might make great classifications, but sometimes when we're doing biological research, we're interested in sort of what the underlying mechanism is and not just getting the right answer all the time. And naive Bayes can be very useful for sort of seeing why that decision was made. And so I find it's a pretty useful method in that context. So it really depends on what you want to accomplish.
So Josh, here's a question from my side. Recently, in the Microsoft Imagine Cup regional finals here in Pakistan, I and my group mates we pitched an idea which was truly based on nutrigenomics. The idea was like we really merged nutrigenomics, nutrigenetics, and pharmacodynamics in a single whole lot project. So the workflow was that we are gonna take the different the biomarkers like the whole blood picture test and cardiovascular observations from the clinic, and then we will be taking out the data set and training our models on that, and base on the basis of the standards we are gonna uh, take it to the next level. So but as you know that this field is not as much possible to implement, so we were not able to implement it in a very smooth manner. So can you please elaborate that how these sort of projects can literally be able to have in real world?
So you're asking me about implementation details. I mean, it just really depends on your data and whatnot. Yeah, I mean, one of the great struggles of machine learning, data science, bioinformatics, whatever you want to call it, one of the great struggles that we have, and eternal struggles, is dealing with the data, being in different formats. Sometimes trying to combine different data sets is tricky because they've got different identifiers. Um, a lot of work ends up being just data, what they call data wrangling or data massaging, or I don't know. There's lots of terms for it. It's what we spend most of our time doing. And actually, I can share. Let me see if I can share you. I've actually got code that implements um, this this thing that I was describing. Let me hold on. Let me share this window with you so you can see how much of the code is dedicated to. Oops. Open this, and we're going to share a window. I'm going to share this. Let me show you. So this is I decided to do this in R. And so, you know, you got in R, we import some libraries. If this was Python, we'd be importing packages. So I import the data, and this is one file that I have to import from, and I have to do. I'm already doing a lot of massaging, trying to get the data to work, and then I have to merge it with a different data set. And so this is the part that works about with merging. So this is the code involved in merging. So there's a lot of code. And we're still just merging the data at this point. We're still just merging. And at this point, we finally merged the two files that we need. Okay. We haven't actually done any machine learning at this point. Now we're just creating the active and inactive genes. So we're doing that. Now we're just doing some simple visualizations to confirm whether or not a naive Bayes will apply. So in this example, I used 5 different histone modifications to determine whether or not a gene was active or not. And I'm using this histogram to determine whether or not there's differences enough to see. So it looks like, so if the salmon color is for active genes and the teal color, this greenish or bluish, is for inactive genes, we can see that this histone modification is biased. And that bias means that naive Bayes will probably work well. Okay, so we finally get down to applying naive Bayes. And here it is. This is it. This is the 2 lines. This is the actual machine learning. 2 lines of code right here. That's all there was in this entire file. Everything else was just dealing with the data not being in the right format and being easy to use. And then we do our confusion matrix, and actually it actually worked pretty well. We had 81% accuracy. So it actually worked pretty well. But the actual data science or sort of a machine learning aspect of this is just these 2 lines. The good news is that if we wanted to try deep learning or a neural network, or we wanted to use support vector machines, all we would have to do is change these 2 lines, and then we could do it with a different machine learning method, and we could compare the results and see which one works better. Um, but you see that, like, you know, applying machine learning in biological realms is pretty tricky. There's a lot of data wrangling that has to be done, and sometimes it helps to have expert knowledge on how that data was generated and sort of what to expect and how it can fit together with other pieces of data.
Well, Joshua, got it. That was really a great elaboration. Attendees, if you folks have any questions, you can enlist that in chat.
Someone asked if we can use another algorithm in machine learning rather than naive Bayes, and the answer is yes. You can use whatever method you want to use, as long as it applies to, some methods are very good for sort of discrete outcomes, like active, not active, and some are better for sort of regression style or continuous outcomes. And so just make sure that you're using a machine learning method that's good for classifying rather than sort of predicting a continuous value. But yeah, you can just throw whatever machine learning method you want and see how it works. Any other questions?
Well, Josh, here's a question from Angela. You can answer it first.
So yeah, so how do we decide when I need to use a classical machine learning algorithms or neural networks? To be honest, I actually think that's kind of funny terminology that you would call classical machine learnings versus neural networks, because neural networks have been around for 60 years. They're as classical as pretty much any other method out there. They actually predate support vector machines, and they predate like XGBoost and a lot of like other machine learning methods, or random forests and things like that. They were invented in the 1950s. So anyways, how do you decide when to use a classical machine learning algorithm or neural networks or something? Like I said, it depends on what you want. Do you just want answers, or do you want insight into how those answers were generated? So you have to answer those questions. The other question is just performance based. What I like to do is, you pick a handful of methods. You could do neural networks. Neural networks, by the way, are not always the best. I mean, they work well, but they're not always the best. And so pick a few methods that you like. You might like XGBoost, you might like neural networks, you might like support vector machines. We can try all 3 and see how well they perform and sort of evaluate how well they do. And it could be that neural networks is not the top choice. Neural networks can be finicky and hard to decide how many hidden layers you need or how many activation nodes you need in each hidden layer. There's a lot of like what do they call hyperparameters, or sort of like fudge factors, or things you have to fiddle with manually to get neural networks to work very well. And sometimes that can be hard to do. And so what we do is we can just test things with cross validation and see which one works best for us. And we can just do it objectively. We don't have to be, we don't have to have a lot of domain knowledge to know. We can just try them out and see how they perform.
So Josh, going forward, as you know, nutrigenomics is one of the trendy topics nowadays. That how can we control our diet or personalized diet on the basis of genes, because as they differ person to person. So my question is, how can we get those biomarkers and craft a new data set, because these sort of things are really not accessible to every person.
Yeah, that's a good question. And unfortunately, I'm probably one of the worst people to ask that question. Unfortunately, I worked in, when I worked in genetics, I did cancer genetics. And so I can tell you a little bit about cancer genetics, but I'm no expert on nutrigenomics or even diets in general. So I, you know, I mean, other than say, a lot of people have like allergies or whatever, like you could say maybe we could find a lot of people with certain allergies to certain foods, like eggs. Some people have allergies to eggs, which I love eggs, so that sounds horrible to me. But maybe if we found a lot of people with eggs allergies, we could collect some cells from them, and we could do ChIP-seq, and we could do RNA-seq, and we could identify what genes are active. You could also just sequence the genome and see if they've got some mutations in genes, and we could compare those to people that are not allergic to eggs, and we could say, hey, this mutation that occurs at this specific location in the genome. This occurs in people who have egg allergies, and it does not occur in people who do not have egg allergies. And so maybe we can attribute that to why they're why they have the allergy. But in all likelihood, it's probably more complicated, and that's when machine learning comes in, right? Like I showed you with naive Bayes, instead of just looking at one place, we can look at the whole genome, and we can look at multiple things. We can look at protein binding to the genome. We could look at gene activity. We could have a pretty rich data set entering um, into our machine learning algorithm, and then the output could be you know a not allergic to eggs or is allergic to eggs. And so that would be a way to try to help, rather than saying, here, just try these eggs and see if they make you feel sick. We could use our machine learning algorithm to try to figure that out in advance so that you don't have to feel sick. So that's, I mean, I'll be honest, I don't know much about nutrigenomics, but that's just my guess as to how you might go about doing it.
That's really well elaborated. I really got the concept. Folks, if you folks have any question, you can enlist in the chat. Otherwise, Josh, here's a last question might be
Okay.
that we have a very booming ecosystem of startup culture or entrepreneurship. So how would you guide the youngsters about the startups in healthcare or in this specific domain? What is the scope of startup culture in this domain?
I mean, I think it's a fantastic place to be. I can't really offer advice on specific details, but I'm confident that it's a place that's, people get sick all over the world. Healthcare is a place that people will spend money to try to improve. Right, feeling good, feeling healthy is one of the most valuable things to us. It's more valuable than money, for me at least. I'd rather be healthy and poor than very sick and very wealthy. Right? You don't. That just sounds like misery. And so I think healthcare is a fantastic place to be, and I think healthcare is now generating crazy amounts of data. These ChIP-seq data sets that I was talking about are just the tip of the iceberg in terms of these massive data sets. And so big data, being able to handle big data, being able to apply machine learning to big data. These are all skills that I think are going to be incredibly useful and helpful and marketable skills, you know. So I think it's super exciting that's something that's going on where you are. That's yeah, there's going to be a lot of cool stuff, I think, happening in the next 10, 15 years. And you guys are going to be right in the middle of that, probably, you know, selling off some stock options and becoming very wealthy. So that sounds exciting. Anyways, yeah, that's probably the last question. I've got to go. But I really appreciate you guys having me here and allowing me to speak and being patient and listening to me. Thank you very much.
Thanks for your time. Over to Angie. Angie is just gonna wrap up the event.
Okay.
It was a great session. On behalf of myself and my all organizer friends and attendees, I want to thank you for insightful presentation. We are grateful for the time and effort you took to share your knowledge and experience with us. Thank you for your valuable contribution. I also want to say thanks to attendees. We hope you had fun and we look forward to seeing you at next events. We will provide you the certification for those who fill out the form, so don't forget to fill out form. Many thanks from all of us.
Thank you very much. Bye.
Thanks, Joshua. We will soon invite you again because we have a lot of questions to ask, but the time is really limited and we have some time constraints as well. We are really thankful for your time, and that's all. Signing off for the event. We'll see you next time.
Okay. Bye.
Thank you. Bye-bye.